Hello, this is a negative strand RNA viruses tutorial presented by the SchrDbb, also called Dassi Bopda Blondel reference Lippincott illustrated review of microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So we are going to start our tutorial of negative strand RNA viruses with the classification of the negative strand RNA viruses. So we start by knowing that RNA viruses divided into two single-stranded, double-stranded. In single-stranded, you're going to have negative strand and positive stranded. In negative strand, they are in 3' to 5' direction of RNA, and they are in 5' to 3' direction of RNA. That's positive stranded RNA, and positive stranded DNA are 3' to 5' direction. So for you to understand better on this, you're going to just go on the tutorial that we've seen initially on the introduction of virology. And for you to understand again best of it, go on the tutorial on genetics that we did. You go on the genetics playlist and see introduction of genetics and the genomic review. So we have on the negative strand helical enveloped viruses, we have Rubula Virus. That's one. We have Paramyxoviridae. The Rubula Virus is made up of the Lissa virus. The Lissa virus is an example is rabies virus. Is it clear? So Lissa virus is rabies virus. That's also the name. We have Paramyxoviridae. The Paramyxoviridae family is made up of the Paramyxoviridae subfamily, which is made up of, and it's made up of the Paramyxoviridae subfamily, and is also made up of the Pneumoviridae subfamily. Now, in the Paramyxoviridae subfamily, you have the Paramyxovirus, which an example is human parainfluenza type 1 and type 3 virus. We have the Rubula Virus, not Rubella. We have already seen Rubella before. We have the Rubula Virus, which is made up of the human parainfluenza type 2 and type 4, and the mumps virus. We have the Mobile virus, which is made up of the measles virus. Now, also the next subfamily is the Pneumoviridae. The Pneumoviridae is made up of the pneumovirus, which is made up of the respiratory syncytial virus. Most of these viruses are going to affect the respiratory system. Then the next one is the Orthomyxoviridae family. Is another family under the negative stranded helical envelope virus RNA viruses. Now we continue under the helical non-envelope RNA viruses. We have also the Filoviridae family, where we have Ebola and the Marburg virus. We have the Bornea Viridae family, where we have the Bornea virus. Example, the California Encephalitis virus and Lacrosse Encephalitis virus. We have the Hanta virus, which is made up of the Hanta viruses, and we have the Arena Viridae family, which is made up of the lymphocytic choriomeningitis virus, the Junion virus, the Macupa virus, and the Lassa virus. Now, the surface glycoproteins. So the surface this is just a diagram to show you rabies virus or Lissa virus. Now, the rabies virus is made up of the surface glycoproteins, which are the G protein, which are extending from the lipid envelope. The G protein is antigenic. Is the antigen is the one that is having the antigenic property of the surface glycoprotein. Is having the antigenic property of the rabies. Is it clear? So the G protein is antigenic, resulting in host production of neutralizing antibodies. So now, what is the life cycle of the rabies infection? So what's the pathogenesis? So what happens is that you have a patient, an infected canine family animal, or you can have an infected animal. It can be a bat, it can be any animal from canine family like the dog, it can be a wolf. So it bites you. So when it bites, now what happens is that the rabbit is going to travel through it's not going to travel through blood, but it's going to travel through the nerves. Is it clear? So rabies is going to travel through the nerves. Is it clear? So once these are the different things that can cause the infection. It can be the raccoon, anything from the canine family. It can be fox, it can be raccoon, it can be the skunk, even can be an unvaccinated dog. It can be bats in 10% and others can be in 11%. So when it bites, it goes, it passes through your neuron, your peripheral neuron, and then it's going to following inoculation. The virus may replicate locally, but then enter into the peripheral nervous system, where it's going to be passively travel to the central nervous system. Then later on, the virus is next going to pass through the brainstem, the cerebellum, and other brain structure, and cause diffuse encephalitis. So it's going to pass through other brain structure to cause diffuse encephalitis. Then the last one is that from the brain, the rabies virus can travel now along your autonomic nerves, leading to infection of other tissue, including the skin, the cornea, and the salivary gland. So at the level of the salivary gland, you can have excessive spitting coming from your salivary gland. Is it clear? That's why people having rabies the end form of rabies are going to have salivation from the mouth. Is it clear? Just like dogs. Is it clear? They are going to have autonomic barking activity. They can bark just like dogs. Is it clear? So from that is it. You have the cornea is that they are going to be hydrophobic. From the cornea, every time they see water, they are hydrophobic. They are running away from water. And also the same on the skin, they are hydrophobic. They have irritating sensation to water. Is it clear? So those are the different things. Now, an oval migrate body in the brain that can be shown in human rabies. So this is a case of patient that had rabies that is not more, and then you can see the negri bodies in the brain from the biopsy. So what is the model of paramyxovirus? So the model of paramyxovirus is, so we are finished with the rabies. Now we are paramyxoviridae with paramyxovirus. So this is an example where you have a capsid inside a so here we are going to have a non-segmented negative strand RNA, and then outside the nucleocapsid you are going to have the envelope. The envelope is made up of largely glycoprotein with the HN proteins. Is it clear? So we need to know that this is actually what is known as flu with the paramyxovirus. When you have these different pox viruses, the pox fever and all that. Is it clear? So in French, la grippe pox porci, la grippe avian, those avian flu and then pox flu. Is it clear? Because of these paramyxoviruses, and there are two main types of proteins that are antigenic in paramyxovirus, which are the HN or the H proteins. Is it clear? And they are made up of helical nucleocapsid. There are also smaller glycoprotein called the fusion protein. Is it clear? So this is another diagram. So in the past, this avian flu and this pork flu has caused a very great disease. In the 2010, if I'm not mistaken, it has caused a very great worldwide disease where avians such as birds and even chickens were not sold again in the market, because when they were not well cooked, they were resulting to this disease. It was affecting the respiratory tract and making that people were having difficulty in breathing and death in some period in time. But now what is more recent is coronavirus, which is not a foodborne disease, but is more of an air droplet infection, airborne disease. Now this is in the case of mumps. Mumps is going to result to more swelling of the different part of the body, and then mumps usually is going to cause a unilateral is going to cause bilateral swelling more than unilateral swelling. Is it clear? And patients having mumps are also going to have orchitis. So try with mumps showing Barrett's gland swelling. Generally with mumps you have bilateral swelling of the different parts. So these are the Koplik spots. The Koplik spots which are seen in the case of the different phase of measles. You need to know that measles have three main phases in its pathogenesis. The first phase is the prodromal phase. The second phase is the enanthem phase, and the third phase is the exanthem phase. Is it clear? So the prodromal phase of measles is when it's also a period of coryza, where you have fever, you have cough, you have catarrh, you have tearing from the eyes and all that. So that's the prodromal phase of measles. Then you have the enanthem phase of measles, is when you have the coplic spot appearance. So these are the whitish spots inside. So these spots like that, which are whitish inside the mouth, are just known as the coplic spot appearance in the child. And then you have the exanthem phase, which is when the rash appear at the first start at the level of the back of the head. You first have a rash that appear at the back of the head and then spread through the body, and the rash are more like macules. They are maculopapular rash. They are more macules than papules, as compared to chickenpox, which are more papules and vesicles. Papulovesicular. So chickenpox is more papulovesicular, papules and vesicles, and measles is more maculopapular, macules and papules. So now the question you want to ask me is, what are macules? Macules are flat skin rashes which are less than 1 centimeter. Papules are flat are raised skin rashes which has less than 1 centimeter, and vesicle are fluid containing skin rashes which are less than 1 centimeter. So those are the definition of each of them. Papule, which are raised rashes which are less than 1 centimeter. Macules are flat skin rashes less than 1 centimeter, and vesicles are fluid-filled rashes less than 1 centimeter. Now the contrary of each of them have their contrary. Example, the contrary of macule is a patch. A patch is a flat rash, which is the skin rash which is greater than 1 centimeter. In this case, a patch is greater than 1 centimeter, but it's still flat skin rash, just like a macule. A papule, the contrary of papule is a plaque or a nodule. Is it clear? A plaque is a raised skin rash of greater than 1 centimeter, just like a papule. Is it clear? And a nodule too is a raised skin rash greater than 1 centimeter, just like a papule. Now, but the difference between a plaque and a papule is that the plaque has a flat top, and a nodule has a dome-shaped top. Is it clear? Now, the last one is the contrary of a vesicle is now the contrary of a vesicle is a bulle, where a bulle is a fluid containing skin rash which is greater than 1 centimeter. And now the last one that I didn't speak about is a pustule, and its contrary, which is abscess. A pustule is a raised skin rash containing pus of less than 1 centimeter, while an abscess is a raised skin rash containing pus of greater than 1 centimeter. So these are the different clinical semiologic terms for integumentary system. So this is the different phases of the measles virus. So we have an incubation period, then we have a viremia. So when you have the primary viremia, that you have the prodromal syndromes. When you have now the secondary viremia, that you can have after the respiratory virus in respiratory tract and the urine, now you have coplic spots and all that. You have the rash, you have the post-infectious encephalomyelitis. The post-infectious encephalomyelitis occur in very rare cases and occur after 5 to 7 years after the infection has been spread. And generally, after the infection, you have virus-specific antibodies are present. Now, the influenza virus, you have electron micrograph. Is it clear? Influenza virus. So this is the influenza virus. So you need to know that with the influenza virus, you have the capsid and you have the envelope. So the envelope now is made up of the hemagglutinin and the neuraminidase. Is it clear? Just like the paramyxovirus and in the case of avian flu. So this is the influenza virus, which is made up of the hemagglutinin and the neuraminidase antigen. Is it clear? And this antigen is what is going to account for the virulence of the influenza virus in case of the avian flu and all that. So now here this diagram is going to show you a cap snatching of the influenza virus. So what happened is that the virus P protein, is it clear? P protein cleave at the 5' terminal sequence. So sequences 10 to 13 amino acid long of the host cell mRNA. So what happens is that the influenza virus actually is an RNA virus, is it clear? And an RNA virus from 5' to 3', and this 5' has a cap, is it clear? And this is a capped host cell mRNA. So this is going to be, so with the mRNA of the total mRNA of the influenza virus, there is only an addition of this particular cap like that in the normal host mRNA, is it clear? So when this cap is removed, this can be used now in it can be used in transcription, is it clear? So you have the removal of the cap. So you have oligonucleotide fragment are then used as a primer synthesis for the different mRNA, is it clear? Because this one is 5' to 3', so now it's going to be converted to produce now the different sequences of the positive viral RNA, and the positive viral RNA can now be used in translation. Now this is now the cause of the influenza virus. So cause of influenza virus is still, you have the prodromal symptom, you have the catarrhal phase, you have the phase now of the disease, and then you have the convalescence, is it clear? So with the prodromal symptoms, you have this thing with the first have the incubation period, then the prodromal symptoms, then you have the virus in the respiratory secretion, you have interferon induction, you have clinical symptoms now in the prodromal, and then the disease period, and then you have the rise in specific antibodies in the convalescent phase. So this is the mechanism of the antigenic shift in influenza virus. So in antigenic shift in influenza virus, how does it occur? So we have hemagglutinin coded by the RNA segment 4. That's the first thing you need to know. This is why actually what happened is, the question is what you have to ask myself. You want to ask me is, what is antigenic shift? Is it clear? So antigenic shift is a phenomenon in which there is more dramatic change in antigenic property of the H and the N protein. It means that what? It means that actually the antigen, normally like other viruses, when you are infected with a virus to a single time, you are not going to be infected by that same virus a later time again. Example, when you have varicella. Varicella is a single time for life, except you are immunosuppressed, where you are going to have herpes zoster or shingles in future time. Is it clear? But varicella is a single time in life generally, and that's generally with all viruses. Measles, single time in life. Smallpox, single time in life, except SchrDbb. So those are the difference. You are going to have all of them single time in life. But influenza virus, and even in the case of rhinovirus with common cold, you have single common cold time with life. But the problem with common cold is that common cold is having many you have many forms of common cold with different antigens of common cold. Is it clear? Now the question is, why is it that there are many influenza virus? Why is that patient can have influenza one time, a flu-like syndrome, then tomorrow he has again another influenza trait, and then after tomorrow he has again another influenza trait? The reason is because of antigenic shift. It's a phenomenon in which there is a dramatic change in the antigenic property of the hemagglutinin and the neuraminidase antigen more than expected. So how does it occur? So this change occurs because you can have hemagglutinin coded by the RNA segment 4. Is it clear? This is a neuraminidase which is normally coded by the RNA segment 6, and it's all going to produce a human influenza virion. Is it clear? These are the different RNA. You can have 1, 2, 3, 4, 5, 6, 7, and 8 RNA segments. Is it clear? Now, what happens is that you have mixed infection of human and animal influenza virion in the same cell. Is it clear? So you can have certain time where you have mixed infection of animal and human influenza virion in the same cell. So let's say that a human cell is going to be infected by the animal influenza and is infected by the human influenza. Is it clear? So when it's infected by animal influenza and human influenza, you are going to see that you are going to have different RNA. So normally, as you've said into the introduction, you said that the hemagglutinin is coded by RNA4 and it's coded by the neuraminidase is coded by RNA6. Is it clear? Let's say that this segment is a segment of the human influenza variant. Now, this is the animal influenza variant. Is it clear? This is the animal influenza variant with the different RNA in bolds. Here, the RNA which are not in bold, which are in light color, are the RNA of the human. The RNA which are not in bold here are the RNA of the animals. Is it clear? Now, when the animal, the RNA of the influenza, let's say human cell, is it clear, is infected by both the influenza which is normally infecting humans and the influenza which is normally infecting animals, you see that there is a cross match to produce a new influenza virus. Is it clear? You see that one can be, you see that in this case you can come 1 and 2 from the animal genome can cross-link with the 3 and 6 from the human genome, which can cross-link with the 7 and 8 from the animal genome again. You see that this different cross-linkage is going to produce again another assortment that can produce again other hemagglutinin and other neuraminidase antigen. You see that at the beginning, this is the neuraminidase. This is how the structure of the hemagglutinin of the human was resembling, and this is how the hemagglutinin of the neuraminidase of the human normal influenza virus resemble. Now this is now the neuraminidase of the normal animal influenza virus resemble, and this is the normal neuraminidase of the animal also. Is it clear? Now when you cross-link it, you see that you have now the hemagglutinin of the human, you have the hemagglutinin of the human, and you have the neuraminidase of the animal. So this one is another influenza virus. So that's why this different assortment can now result to changes. Is it clear? So you have different assortment that result to changes in the different modification of the viral particle. So this is a timeline showing the occurrence of the major outbreak of antigenic shift. So we had one in the 1990s. You had the flu symptoms like avian flu, we have porcine flu, all those were influenza viruses. So one was in 19s, one was in the over the 20 million death worldwide. Is it clear? Was Spanish influenza. One was in 1932. One is in, so this is the H1N1, this is the H1N2, which has caused that. Then this is the Asian influenza, the H2N2, all that is still the hemagglutinin and the neuraminidase. Here you see that the hemagglutinin and neuraminidase, the receptor has been shown, which was H1N1, and then later on, after thinking that the receptor has been known, in 1947 that same receptor has changed in modification, H1H1 has been affected. Now another outbreak has come again, which was Asian, and has killed many people because he has changed in form. The hemagglutinin now has become 2 and the neuraminidase has become 2. Is it clear? The Hong Kong influenza has come again and killed many people, and because he has changed again in form, because the hemagglutinin has become 3 and the neuraminidase has become 2, and they have forgotten that in Central Africa there was the porcine and the avian flu that had occurred in something like 2009-2010, which has killed many people again in Central Africa. And because of the new trend, which is the H3 and N3, hemagglutinin 3 and the neuraminidase 3 antigen substrate, it has killed many people in Central Africa, including Cameroon. in our country. Now that is the case involved there. Now this is the electron microgovernor of Ebola virus. Ebola virus is also another virus that has affected most of Central African country, but not Cameroon in this case, because Cameroon government has highly regulated the transfer of material from one country to another. Now, the next is the transmission of California Encephalitis virus. So it starts by the virus reversibly transmitted between rodent reservoir and mosquitoes. Is it clear? So the mosquito, the reservoir is actually the rodent, and then the mosquito, which is the Aedes trisoriatus mosquito, is the one that is the vector. So the Aedes trisoriatus is now going to, so you have mosquito pass the virus from adult female to their offspring. So the mosquito can pass to the rodent, and the mosquito can even pass to their offspring, to their larva, which can go. And then when the mosquito now is going to pass their virus to human by biting them, that's when you are going to have the California Encephalitis virus. Is it clear? So from here we are finished with all the life cycle involved with the Retroviridae family, all the life cycle involved with this section. Say thanks for your kind attention. Please don't forget to like and subscribe for our channel, Science Jamaica.